Dormant bacteria are discovered by the fact that they appear not to be there, but they can be resuscitated such that they then go on to divide under conditions in which originally they wouldn't. And we discovered that in laboratory cultures of a little bacterium called Micrococcus luteus, where we could prove true dormancy, so that all but 1 in 10 in 100,000 of the cells could be made to go dormant, with almost none of them being alive, and you could dilute them so that none of them were alive in a culture, and yet they weren't all dead because we could get them back again. And what we also went on to discover in this case was that the live cells could secrete a wake-up molecule that told the dormant ones that good times were coming and it was worth waking up again. And we actually purified that molecule. It turned out to be a protein, and it was called a resuscitation promotion factor, and is widely distributed throughout a particular clade of bacteria, particular kinds of bacteria, referred to as actinobacteria or high G plus C gram-positive bacteria. And occasionally distributed in other things, but probably not of functional significance there. So dormancy is clearly common in environmental microbiology. So when you look at the microbes in soils, the seawater, rock pools, take what you will, lakes, environmental circumstances, most of the time they are dormant, because if they had anything to eat, they would eat it and grow until they had used it all up. And clearly, and indeed it's been known for years and years, that if you look down a microscope and count the number of bacteria you can see in, say, a sample from soil, you typically see 100 times more than you can persuade to grow on a culture plate. And that was already telling us that most of them are at least not alive or growing. And what were they? And they weren't dead, because under certain circumstances you can indeed resuscitate them, and people have been trying and improving. The other way you can tell they're there is using molecular methods, because every bacterium has a unique sequence of DNA. So you can look at all the sequences and you can count up all the number of different bacteria that are there, particularly using a gene for something called ribosomal DNA, although other ones are possible. And that again is another way of telling you. And typically, again, you see 100 times more bacteria when you count them by their macromolecules than you can persuade to form a colony. So dormancy is very common, and the only question then arises is does it have a clinical relevance? And what our recent papers have been showing is yes, it does, because we can actually see these things down a microscope. All known actinobacteria have at least one homologue or similar protein to the RPF resuscitation promotion factor that we discovered, and occasionally you find gene sequences for these proteins in gram-negative bacteria, and we think that's unusual and happened through horizontal gene transfer, but the functional significance isn't really known. But really, for practical purpose, we'd see them as being confined to these actinobacteria and not really anything else. And it's not biofilms or not biofilms in this case. Although clearly these organisms too can form biofilms. One of the famous kinds of clinically relevant actinobacteria is Mycobacterium tuberculosis, which is well known to go dormant for a long period of time, and other researchers, and we too, my collaborators, have shown the relevance of RPF to the breaking of dormancy in mycobacteria. And it's a general assumption, although it's extremely hard to prove, because mycobacteria live in lungs of infected laboratory animals and people, and seeing precisely what is happening when you've got to look inside a lung is not really feasible. But you can find strains of mycobacteria with different, or you can create strains of mycobacteria with different amounts of and numbers of RPF, and they are more or less infective, and people have shown that. There's a classical experiment going back to the 40s by Salvador Luria, who simply took a suspension of cells in a laboratory and exposed them to nasty ionizing radiation and watched on a logarithmic scale the number that would create a colony as a function of time. And it went down like this on a logarithmic scale until about 1% were left, and then the slope of that line changed dramatically, and those that were left were in fact much more resistant. But the resistance was phenotypic because you could take them out and show that you could reproduce the exact same curve when you grew them all again. So it wasn't that the genetics had changed, there was a subpopulation that was, for practical purposes, having a sleep at the time, and that made them much more resistant. There's also similar experiments going back again to the 40s, starting with a man called Bigger, looking at the fraction of bacteria that evade antibiotics to which they are normally sensitive, and again, this evasion or persistence in the face of antibiotics is a phenotypic effect, because you can take those self-same cells out and then they all are just as sensitive, except for the 1 in a million that isn't. It's actually in your interest to have a subpopulation not growing in case something nasty happens, so that those can survive the something nasty and then get going again. Clearly, of course, they can't just do it all the time, because if they're functionally dormant forever, they're functionally dead. So clearly they have to go in and out of this state of functional dormancy, and indeed that's precisely what happens. And in laboratory cultures, for instance of Escherichia coli, quite a number of the genes are known. If you want to go into a state A and back to a state B and into a state A and back to a state B, the simplest version has gene number 1 causes state A and gene number 2 kills gene number 1, and then you go back to state B. So, and they're called, it's a slightly silly name, toxin-antitoxin pairs, and there are quite a few examples known. So from the molecular point of view, it's a pretty well established set of phenomena now, but they're very different between different organisms, even though macroscopically it's just as easy to say we have these 3 macrostates: alive, dead, functionally, and dormant. So this relates to what I described earlier, where you could go to the environment, find that there were, by observation, 100 times as many microbes as you could plate out. Nowadays, with this sequencing, where you can actually sequence some or all of the genes of these organisms, you could tell whether they are the ones that were known in the culture collections of microbes we knew and loved. And when you have not succeeded even ever creating them in a laboratory growing medium, then they're typically called non-culturable, but it should be better called not yet cultured. A number of organisms that we know today quite well, Legionella, an obvious example, that had never been seen before it came to prominence when it caused Legionnaire's disease, and people tried to grow it, not very successfully, until they realized in this particular case it needed a slightly weird medium containing a lot of cysteine. And in the case of Helicobacter, for which Barry Marshall got the Nobel Prize for discovering its role in ulcers, it was recognized that this organism needed a lot of urea because it had a very active urease, and that created ammonia, which counteracted the acid in the stomach and so on. So a number of organisms have started life as not yet culturable, but then they become culturable. But there are still a lot that have not yet been cultured, but we know they are there from their macromolecular sequences. Well, from an operational point of view, not yet cultured and dormant are indistinguishable because they are there. If they are capable of becoming alive again, they are therefore resuscitable. You just have to find the right medium in which to do it, or it might be a 2-stage process. So when we discovered the RPF wake-up molecule, we first had to add a very weak nutrient broth to coax them back from their state of profound sleep, and then we could add them to the normal medium where they would normally grow, and off they went. And so, and if you look at macro people, if you find someone who, whether for whatever reason, has been starved or starving for a long time, if you give them a big meal, you'll actually kill them. So you have to bring them back to normal physiology and normal eating quite slowly. So this is not unreasonable that it's true for bacteria as well as people. we would normally say if you're looking for an antigen and you only use one, then clearly you're likely to find only one strain. But if you're looking at the DNA level, looking for particular sequences, you might reasonably expect to pick up all of the Borrelias, and so we would recommend to people to look using molecular methods based on ribosomal DNA or other such genes. It is certainly the case that Borrelia is one of the many organisms that can go dormant inside people and hide for a long while and come out again. And the $64 question, of course, is what makes them wake up, beyond the normal ability of the body to deal with them when they're dormant and only small numbers waking up. We know some mechanisms. There's a group, Mark Light, his name is, in Minnesota, has been publishing for a long time showing that noradrenaline, or norepinephrine in the US, can stimulate the growth of Escherichia coli quite dramatically, and we've seen that too, so it's definitely true. That's true for gram negatives. Years ago, we did try that on our gram positive system. Nothing happened, so again, not every organism is the same. And in a particular area that we've been concentrating on, if you look at all the diseases that are chronic inflammatory diseases, they're also diseases in which iron, the metal iron, IRON, metabolism is dysregulated and iron becomes free. Normally, the thing that limits the growth of microbes in people is the lack of free iron. So if you withhold iron, the bugs won't grow. Whereas if it becomes free because you've got something else bad going on, that can unleash the microbes. And we think that's quite a lot of the story as to what's happening. If you do find that you've got microbes in blood and tissues, and if they get to the tissues, they come via the blood. The question then is, how do they get into the blood? The two main sources are definitely known to be the gut, which of course is full of bacteria to the many trillions of numbers and 10 to the power 13 actually, and oral, especially periodontitis. If you have blood around the gums, it's a two-way street. Blood comes out, bugs go in. And that's known, especially in certain diseases. Rheumatoid is one that it's very clear that microbes can come in via the oral microbiome, but it's considered that the leaky gut in various forms is probably the major source, on the grounds that the overwhelmingly vastest number of microbes which are adjacent to a human definitely are living in the gut. Now, LPS is part of the cell wall of gram-negative bacteria and can be shed. There's a gram of LPS in the gut of an average person. Probably nanograms of the stuff are enough to act as potent inflammagens. So it doesn't need many bacteria, or indeed the cell wall component that may come from the gut or be shed from the blood or tissue to start making this inflammation. And we think that that is one of the major causes of it. So yes, it's the bacteria that provide the LPS, but as an intermediary, it's the LPS that is causing the inflammation. There is considerable evidence for every one of the steps, and we've adduced in our reviews the evidence, including some of our own evidence, looking down microscopes and seeing them. What we've done that's different from others is put it all together and put together the iron, the LPS, the dormancy, the particular microbes, the translocation, and all the evidence as a systems biology problem. In fact, I am a systems biologist, and probably that's what we've done. So the novelty is not so much in any individual thing, but bringing it together as a whole story that is self-consistent. In philosophy of science, this is known as coherence. you believe a scientific theory more if the separate bits hang together and there is evidence for them, but they all end up pointing at the same thing. The Gestalt argument is that if you're going to keep creating LPS, that is a well-known inflammagen that causes inflammation, the LPS has got to keep coming from somewhere, and the only place it comes from is bugs. Now, the bugs can divide, but if they were dividing in your blood all the time, you'd be dead in no time at all. And indeed, the body has means to stop you. If every time you cut your finger, you got an infection and died, there wouldn't be anyone left. So clearly, the body has a certain capacity to get rid of bugs it happens to see in the bloodstream, known as the innate immune response, and indeed the not innate immune response too. That normally works to get rid of this stuff. What we believe happens when bugs go dormant is that they go and hide inside cells where the innate immune system can't see them. But of course, if they suddenly start growing and reproducing, then they can shed their LPS, create trouble, go back and hide. One thing that causes bugs to grow in people when normally they don't is the lack of iron turning into a not lack of iron. If your test is based on culture, i.e. the bugs actually dividing and forming a macroscopic colony, or just turning a transparent broth that allows their growth to go turbid, no, you won't see that if these bugs are dormant, because by definition these bugs are not dividing under the normal circumstances in which you would expect them to divide. If you try and detect them by molecular means, then you will see them. And so there is a move, but it's not yet a very pervasive move, towards changing from classical microbiology techniques that rely on the growth or metabolism of the organisms to means for detecting them based on their macromolecules or some other properties that don't require growth. There are methods that combine mass spectrometry and PCR. There are some new mass spectrometric methods that try and detect bugs directly. Anything that actually is detecting the thing itself and not based on the requirement that the thing itself multiplies. These are the novel methods. PCR requires, or in its normal form, requires that you have a particular set of sequences at both ends of what you're doing the experiment on, and they will then light up anything that has those, and it doesn't much matter what's in between. So normally you would pick PCR probes that work for bacteria, then you'd have a different set for fungi, a different set for certain kinds of parasites. There's nothing that would work for everything, because it would wake up your cell, just show your cells too, which is very interesting. So yes, there are PCR methods for different classes of organism, and they're all PCR, but you use different primers to light things up. All of the modern chronic inflammatory diseases, from atherosclerosis, types 1 and 2 diabetes, the neurodegenerative diseases such as Alzheimer's and Parkinson's, autoimmune diseases such as rheumatoid and psoriasis, ankylosing spondylitis. All of these are, if you look at the right literature, have well-established microbial components to them. There's a nice story about rheumatoid and Proteus by a man called Alan Ebringer from King's College London, and he essentially shows, starting from the recognition that 5 times as many women get it as men, which makes it hard to explain in a sort of conventional genetic way. And according to him, and I think a lot of evidence, if you have a urinary tract infection, which women are much more likely to get, these Proteus have a particular antigen, and you can find antibodies to that antigen which are known also to be antibodies to the things that are attacked in rheumatoid arthritis. And it's almost beyond happenstance that this is essentially the cause. There's a science joke that goes something like, when a thing was new, people said it was not true. When its truth could not be denied, people said it was not important. And when its importance could not be denied, people said it was not new. Or the short version of that goes, it's rubbish, it doesn't matter, we knew it all along. We are somewhere between stages 1 and 2 of that at the moment, in my opinion. And clearly we need to do more experiments to persuade more people, if that's what people wish to be persuaded, if they're interested in the scientific truth, that there is a big story here that is significant from a medical point of view, and that, you know, the normal recognition is that if you understand a complex biological process better and it's a pathological one, understanding it will lead you to treatments and cures that will, or in alleviations that will help patients. I'm not a medical doctor, but that's the normal assumption that we all follow.